You know how scientists have been mapping the human genome like for years?
Yeah.
Well, it turns out,
Mm-hmm.
they've been missing like half the picture.
Uh-oh.
And we're talking about silencers, which are the genetic off switches.
Right.
And they're just as important as those on switches, the ones that we call enhancers.
Absolutely.
So today we're taking a deep dive into this groundbreaking study
Okay.
that was published in Molecular Cell, and it's all about these underappreciated silencers.
It's true. Silencers have definitely been kind of overshadowed by enhancers for a long time. You see,
Yeah.
every cell in our body, right? It contains the same DNA,
Okay.
our entire genetic blueprint.
Yeah.
But different cells need to express different genes to do their jobs.
Right.
And that's where gene regulation comes in. It's like a complex control panel that determines which genes get turned on and off in each cell.
So if gene regulation is the control panel,
Yes.
then enhancers and silencers are like the individual switches.
Exactly. Enhancers boost the expression of certain genes,
Okay.
while silencers do the opposite.
Gotcha.
They repress gene activity,
Wow.
making sure that certain genes stay turned off when they need to.
Okay, that makes sense.
Yeah.
But if silencers are so important,
Yeah.
why haven't we heard more about them? Why have they been flying under the radar for so long?
Well, for a long time, scientists kind of assumed that silencers were just the mirror image of enhancers, like a photographic negative.
Oh,
But
okay.
as this new research shows,
Yeah.
silencers are much more complex and fascinating
Right.
than we initially thought.
This is where that silencer-seq technique comes in, right?
Yes.
The one developed by the Stark Lab.
That's the one. And they were actually able to identify over 800 silencers in the fruit fly genome using this method.
Wow. 800 silencers.
Oh.
That's incredible. So how does this silencer-seq method actually work?
Oh, it's really clever.
Do they just like go through the genome line by line searching for these hidden silencers?
Not quite. They cleverly designed a system to kind of trap silencers by pairing them with their nemesis, a powerful enhancer.
Okay.
So they created a library of DNA fragments and paired each fragment with an enhancer.
Gotcha.
If the fragment contained a silencer, it would essentially sabotage the enhancer's activity,
Interesting.
preventing the gene from turning on.
So by looking for those fragments that reduce gene activity, they were able to zero in on the silencers hiding within the genome. That's brilliant.
It is. It's like setting a trap and seeing what you catch.
Yeah.
And what they caught was a treasure trove of information about silencers.
Okay, I'm hooked. Tell me more about what they discovered about these silencers.
Okay.
What secrets did they uncover?
Well, one exciting finding
Yeah.
was the discovery of specific DNA sequences
Okay.
called motifs that are frequently found within silencers.
Okay.
These motifs act like landing pads for transcription factors,
Okay.
which are proteins that control gene expression.
So these motifs are like little flags
Yes.
that signal to specific transcription factors.
Right.
Hey, come bind here and help turn this gene off.
Exactly. And they found 3 of these motifs,
Wow.
one of which, called DLM3, had been predicted years ago, but had never been experimentally verified until now.
Oh, wow.
This discovery alone is a huge step forward in understanding how silencers work.
And this is where things get even more interesting, right?
Yes.
They actually discovered a whole new transcription factor connected to this DLM3
Right.
motif. What was it called again?
They named it SAFT, short for silencer-associated factor.
Okay.
And it seems to play a vital role in keeping genes turned off in specific tissues, like the brain and ovaries of fruit flies.
Okay.
This is particularly important for development and fertility.
Gotcha.
Without SAFT doing its job, things can go awry.
So this is fascinating for understanding fruit flies.
Yeah.
But what does this tell us about our own genes?
Right.
Could similar mechanisms be at play in humans?
That's a great question.
Yeah.
And the answer is likely yes. While we don't have an exact equivalent of SAFT in humans,
Okay.
we do have similar transcription factors and DNA motifs that play a role in gene silencing.
Right.
And remember,
Yeah.
the fundamental principles of gene regulation are remarkably conserved across species.
Right.
So what we're learning about silencers in fruit flies probably has parallels in our own cells.
So it's like cracking a code in a simpler organism
Yeah.
can give us clues to unlock the secrets of our own more complex genome.
Precisely. And that's what makes this research so exciting. By understanding how silencers work in fruit flies,
Yeah.
we can start to look for similar elements and mechanisms in humans.
Go.
In fact, there's already evidence suggesting that disruptions in silencer function could be linked to various diseases, even cancer.
Oh, wow.
Imagine if a silencer that's supposed to keep a tumor-suppressing gene turned off malfunctions.
Oh, no.
That could have serious consequences.
Yeah, that makes sense.
Yeah.
Now you mention it. It makes you wonder just how many other secrets are hiding within our DNA.
Right.
It seems like we're just beginning to understand the true complexity of gene regulation.
Absolutely. This research is really just the tip of the iceberg.
Yeah.
There's so much more to explore and understand about silencers, their functions,
Right.
and their potential roles in human health.
So you've got these silencers,
Right.
these genetic off switches,
Yeah.
and now we're starting to uncover their secrets.
Yeah.
But how do they actually work?
Right.
What makes them tick? We'll dive straight into those details right after this.
Sounds good.
We were just talking about how silencers could play a role in diseases like cancer.
Right.
And that really got me thinking. It's like, if these off switches malfunction,
Yeah.
things can go haywire.
Exactly. And remember those 3 DNA motifs we discussed earlier?
Yeah.
Those landing pads for transcription factors? Well, it turns out one of them is particularly interesting.
Okay.
It's called DLM3.
DLM3.
Yeah.
Okay.
And it's closely associated with SOFT,
Okay.
that unique transcription factor we talk about.
Right. SAFT, the one that can navigate those densely packed regions of DNA.
Yes.
Kind of like finding a parking spot in a crowded garage.
That's a great analogy. And the Stark Labs research found that SAFT actually binds to this DLM3 motif and plays a key role in silencing specific genes in fruit flies.
Okay.
In fact, they discovered that SAFT is particularly active in the brain and ovaries,
Interesting.
suggesting it's essential for normal development and fertility.
Okay, so SAFT binds to this DLM3 motifs.
Yeah.
And together they act like a powerful silencing duo.
Right.
But how does SAFT actually manage to find its target in such a crowded environment? Is it like a genetic GPS or something?
It's not quite GPS,
Okay.
but it does seem to have a remarkable ability to target specific DNA sequences,
Right.
even when the DNA is tightly packed. And this is one of the reasons why silencers have been so elusive for so long. They're often hidden within these densely packed regions, making them difficult to detect with traditional methods.
So SAFT is like a master key that can unlock the silencing power of the DLM3 motif,
Exactly.
even in the most crowded corners of the genome.
Yeah.
And we know this is crucial for fruit flies.
Right.
But what about humans?
Right.
Do we have anything similar to SAFT or DLM3?
Well, while we don't have exact equivalents of SAFT and DLM3 in humans, we do have similar transcription factors and DNA motifs that play a role in gene silencing. And remember, the fundamental principles of gene regulation are remarkably conserved across species.
Right, right.
So what we're learning about silencers and fruit flies can give us valuable insights into how gene silencing works in humans.
So it's like studying a simpler model can help us understand the more complex workings of our own genome.
Exactly.
That makes a lot of sense.
And the more we understand about silencers, the better equipped we'll be to tackle diseases that might be linked to disruptions in gene silencing.
This is starting to feel like we're on the verge of a whole new frontier in genetics. We've been so focused on the genes themselves and what turns them on.
Yeah.
But now we're realizing that the off switches are just as important.
Absolutely.
Maybe even more so.
It's a real paradigm shift. And think about it.
Yeah.
If we can figure out how to manipulate silencers,
Right.
we might be able to develop new therapies for a wide range of diseases.
That's an incredible thought. We could potentially target specific silencers to turn off harmful genes or even reactivate beneficial genes that have been silenced.
The possibilities are truly exciting. And that's why research like the Stark Labs is so important.
Yeah.
They're paving the way for a whole new wave of discoveries in this field.
Well, this deep dive has certainly opened my eyes to the fascinating world of silencers.
Good.
It's like we've been exploring a vast genetic landscape and just discovered a whole new continent.
A whole new continent. I like that.
But we've only just begun to explore this new territory.
Right.
What other mysteries might be lurking within these silencers? We'll find out more about that after this.
So we've been talking about like all this potential, for manipulating silencers for like therapeutic purposes.
Yes.
And that's a really exciting prospect, but it also kind of raises some questions. If we start tinkering with these genetic off switches,
yes,
could there be like unintended consequences?
That's a crucial point to consider.
Right.
Anytime we're dealing with something as fundamental as gene regulation, we need to proceed with caution.
Right.
But the potential benefits are so significant that it would be, almost irresponsible not to explore this avenue further.
Absolutely. It's all about finding that balance.
Right.
Between pushing the boundaries of science and making sure we're not messing things up.
Exactly.
Yeah.
And that's why research like the Stark Labs is so valuable.
By meticulously mapping out silencers and understanding how they work, we're laying the groundwork for future therapies that could be incredibly precise and targeted.
So this isn't just about finding silencers.
Right.
It's about understanding their connections and interactions within this complex web of gene regulation.
Absolutely.
It's we need to learn the language of silencers before we can write a symphony.
That's a beautiful analogy. And it highlights, the importance of continued research in this field. We've only just scratched the surface of the silencer landscape.
Right.
And there are undoubtedly many more discoveries waiting to be made.
Well, this deep dive has been really amazing for me.
Oh, good. I'm glad.
I had no idea just how much was going on, beneath the surface of our DNA.
Right.
It's like we've been reading a book and suddenly realize there's a whole secret chapter hidden between the lines.
And who knows what other hidden chapters are waiting to be discovered. That's the beauty of science.
Yeah.
It's a constant journey of exploration and discovery.
And on that note, we've been talking about silencers in the context of disease.
Right.
But they're also essential for normal development.
Absolutely.
Think about it, from the moment of conception, our genes are being meticulously orchestrated by this complex interplay of enhancers and silencers.
It's amazing.
Ensuring that cells differentiate and tissues form in the right place at the right time.
Right.
This is a really remarkable process.
It is.
And silencers play a crucial role in this genetic symphony of life.
It really is an incredible feat of molecular choreography.
So keep those questions coming. Stay curious.
Yes.
And who knows, maybe you'll be the one to uncover the next big breakthrough in the world of silencers.